Great, and welcome. We might give a couple more people time to join us as well. If you did pre-register and were able to get a free student modeling pack, if you could type in the chat or give us a thumbs up. And if you didn't, don't worry, we will be sending packs to anyone who requests one. We'll get a link to that in the chat in just a couple minutes. All right, I think, there we go. We've got a couple more people joining us. Welcome, Aaron and Hector. Great. All right, well, I think we'll get started. I just want to welcome everyone to exploring the wonderful world of cell membranes, proteins, lipids, and ions. Oh my. My name is Chris Herman, and I'll be the moderator today. We have a couple people who are supporting us behind the scenes. We've got Heather and Brian, and Chris Chou will be our presenter for the next 75 minutes. If you wouldn't mind to just go ahead and use that chat to communicate during the sessions, and Heather will post links to resources and registration for your free student modeling pack. If you pre-registered and you want to let us know in the chat that you received a free student modeling pack. If not, no worries. We will be posting a link so that you can request one, and we'll send that out to you. I'll also help Chris to monitor the chat for any questions. And now I have the privilege of introducing 3D Molecular Design's model teacher, Chris Chou. Chris holds a bachelor's in bioengineering and a master's in curriculum and instruction from the University of Illinois at Urbana-Champaign. She has been a science educator for 20 years and enjoys facilitating workshops for science teachers in her district and at national conferences. She received the 2014 Spotlight Award for Excellence in Teaching from the St. Vrain Valley Education Foundation and the 2016 Outstanding Biology Teacher Award in Colorado from NABT. She currently teaches AP Biology, Biotechnology, and coordinates the Medical and Bioscience Academy program at Longmont High School in Colorado. Now let's begin the program. Take it away, Chris.
Hi, welcome everybody. Good afternoon to those of you in California. It is more like evening here in Colorado where I am at. All right, so thank you for joining us. If you wouldn't mind in the chat, feel free to go ahead and type in where you're from and what you teach and where you're at. I would love to know whether you're middle school teachers, high school teachers, and what grade levels you teach. So I teach in Longmont, Colorado, and mostly AP biology and freshman biology, as well as biotech. So this kit we're going to talk about today and the way I present this information is actually pretty much how it goes in my classroom. So I'm glad you joined us today. All right, so our goals today are to model the structure and functions of the cell membrane. We're going to explore using this kit how we can demonstrate the role of ions and water and how they are transported through transport proteins across the cell membrane. And then as sort of a applications piece, we're going to be discussing the Cftr gene as well as the protein and how problems with this gene can cause and lead to cystic fibrosis. So I wanted to share with you a little bit of why I'm along this modeling journey. These are photos that you see of students in my AP biology class using the very kits that 3D Molecular Designs sells. And it's fun and engaging for my students. One of the most important practices of science is to model things. And so it's really important to have tactile objects. And so these kits do a really great job of having these tactile objects to be able for students to be able to see things, move things, discuss about their ideas, to reveal misconceptions, and so it's just a great way to also encourage communication and collaboration in my classroom. These photos are pre-COVID, clearly. They don't have masks on. If I were to put up the new photos I've taken this year, everyone's got their masks on. So that's the environment we're in. Okay, so if you pre-registered for this particular workshop and have that kit with you at home, these are some of the items that you have in your kit. This is the student modeling pack. There's also classroom kits available with, larger things and more objects, and great for groups of like 4 to 6. So I have quite a few of those. Last year, when I was virtual during the time I taught about cell membranes with my biology class, I actually sent home a student modeling pack with every single one of my students so that they could model at home with me while I did it on camera, just like I am with you guys today. So as we look at kind of keeping an eye on chat here, seeing that there are folks from a variety of places in California. We've got Joy with honors biology and CP level. We've got some homeschooling, and Ryan is a CSU Sacramento student. All right, great. So, and pre-service teachers, we love that. Okay, so I think you would probably hit a lesson on cell membranes out of the ballpark if you introduce this to your students. So I think it's a great way to be able to demonstrate your teaching skills, your knowledge with having students interact with these models. So, okay, so this kit you have, if you have it, you'll want to assemble some things and get out some scissors and glue and tape to put together some items shown here. And if you don't have time to do it, or you haven't got one of the kits yet, there's instructions that are part of it. And then you can kind of just watch and observe how I use the models, because I'm going to have it on my camera here as well. So you can see that there. Couple of things, if you're seeing the camera with the camera view, the items in the student modeling pack don't have any coloration on it. And so these little things are representing water molecules. What I like to do is just to have students get a red marker and just color it so that they have the oxygen there in the middle is colored red. And then what's also really helpful with these purple squares and blue dots is that in the actual classroom kit, they don't come labeled as well. So you could just have students, it's a great student aid job, is just to have them label. The blue dots represent sodium ions and the purple squares represent potassium ions. So if you have students write that on there, it's a little bit easier to figure out which one is which, especially when we're modeling sodium and potassium pumps and the transport of these ions. Okay, so let's go ahead and start with using the kit. And if you don't have one, no problems. I would love participant engagement here. So feel free to type in the chat as we're looking at these 3 different models or representations of phospholipids. What do you notice and what do you wonder? So feel free to type in the chat. These are representations of phospholipids. What are some things that you notice about these 3 representations? Okay, NH says they have 2 tails. Excellent. There are different levels of detail. Thank you, Ryan. Okay, students often will talk about the colors that they see and maybe some of the things they wonder, especially if their chemistry background is still developing or they haven't taken chemistry yet. They might be wondering what the different colors represent, the red versus the blue versus yellow. And so those are some details that we can fill in as we go along with this lesson. Okay, so here is a representation of phospholipid molecule. And at the very top, I kind of think this looks like a little bit like a funny looking giraffe with the long legs and the short neck. And so the top part, the head part here, is where there's a phosphate group. So in this diagram, that phosphate is colored as yellow, and oxygen molecules are red, or atoms are red, and then black is carbon, and these are blue here, but in a lot of the other diagrams, they show as white. So those are hydrogen and carbon tails that we see there. And with the top part, it's the kind of the phosphate group is the prominent feature up top. So the top half of the molecule with the phosphate group is polar, and it's attracted to water, so it's hydrophilic. And then these two tails that we see down here are composed of carbons and hydrogens, so they're hydrocarbon tails, and they repel water, so they are non-polar or hydrophobic. So we do spend time talking about these two different properties of molecules, whether they're hydrophilic or hydrophobic, because that's going to dictate how they behave in water and other types of environments. Okay, so if you were to put phospholipids, you can have students pull out a graphic that looks like this with their kit, or you can print it out. If you had a big beaker of water and dropped some phospholipids into this beaker of water, how might these phospholipids arrange in a watery environment? Understanding that part of the molecule is hydrophilic and are attracted to water, and part of the molecule is hydrophobic, repelling water. What are some ways that you think these phospholipids can be arranged? So if you're in the chat, feel free to type some of your thoughts. How might these be arranged? And so in the classroom, you could have students take their phospholipids and arrange them on their desks and come up with some different ways they might arrange things, and then have them explain why. Okay, so we have Ryan mentioning a sphere with tails on the inside. Okay, excellent. And H, all the heads would be towards the outside and the tails inside. Excellent. Okay, so here's a variety of ways that these phospholipids can be arranged. And so I've got a couple of them here shown in my camera view here. So they can be arranged in a small bilayer like sphere called a liposome. You could also have a micelle, which is a very small, tiny little droplets there of lipids, or you can have a sheet of them as the traditional, what we've seen with cell membranes, that bilayer and the phospholipids going across. So a variety of ways. I was doing some research into the delivery vehicle for the mRNA vaccines, and they're also lipid based, but different than these phospholipids. So it's kind of interesting. You can kind of mention that, how the delivery vehicle for all of these COVID vaccines are also lipids as well. And you can talk about some of the properties that they had to design these lipids so that they would be able to travel safely through the bloodstream and then be able to fuse with cell membranes to be able to drop off the mRNA into the cells. Okay, so cell membranes are a phospholipid bilayer, and you can see two layers where the tails are facing the inside, as NH mentioned, tails towards the inside away from water. And these phospholipid heads, there's the extracellular layer of these phospholipids and the intracellular layer, and these phospholipids are touching the water. And so anything, so you can see that the tails in the middle are these hydrophobic barrier, and the hydrophilic heads kind of pieced together. So this graphic here is showing a couple of models that are sold by 3D Molecular Designs, which are kind of neat to have these tactile models to use, or some of you with a 3D printer out there could find some files for them and actually print them out on your 3D printers as well. So they're kind of handy to have any of these models that you can get. Okay, so let's take a look at a animation of a phospholipid bilayer of the cell membrane. So I like showing this animation because it helps students to see that the traditional 2D flat textbook picture of phospholipids doesn't really emphasize the fact that there's fluidity with the membrane and how these molecules are constantly moving and pushing and bumping against each other, and that these hydrophobic interactions of the tails are keeping that lipid bilayer together. And the other thing is that these phospholipids, they can move laterally side to side. So I'm going to play this again. And so I think of it like an ocean, the surface of an ocean. So it's definitely not static, and it allows you to be able to tell students how these phospholipids are moving. Sometimes there are gaps between them that might allow certain molecules to kind of slip past these phospholipids, and others, because of the property of the polar heads or the nonpolar tails, don't allow molecules to cross. So besides the phospholipids, there's other components of the cell membrane. There's the cholesterol molecules that are embedded in the middle next to those hydrophobic tails. So again, the properties matching because these cholesterol molecules are also nonpolar. They help these tails to kind of stay together. You also have a variety of proteins scattered throughout the cell membrane. There's just a few showing in this diagram, but there's a lot more happening in our actual cell membrane. So some of them are span the entire length of the cell membrane. So those are integral proteins or transmembrane proteins. And then you have ones that are on the surface of the outside of the cell, extracellular side or the intracellular side. Those are peripheral proteins. They help to anchor the extracellular matrix on the outside or the microfilaments of the cytoskeleton on the inside of the cell. And then you have other proteins that have these green branches. These are carbohydrate groups. And so these are glycoproteins. So you can see there's a variety of different types of molecules that comprises the cell membrane. So you might say it's a mosaic. So the cell membrane is a fluid mosaic model. Okay, so using the student kit or the phospholipids that come individual ones in the classroom kit, you can see this graphic shows how students could show how the phospholipids would be arranged for the phospholipid bilayer. And then I want to replace this with sort of an easier model for the cell membrane. So you can, this one is, they kind of look like railroad tracks, but it can replace all those individual phospholipids, makes it much easier to model the cell membrane. And then you can put different proteins across them to show that there are different transport proteins that are embedded across the cell membrane. And then we can model some different molecules moving across. So this is a diagram of the classroom kit. And if on camera you can see, I've got the student modeling pack. And so students can have their own like cardboard flat background with these different proteins embedded in there as well. To help students, if you're teaching virtually from home, I label them so they can identify the different types of proteins. In the kits, you get stickers for the classroom kits, stickers to put on the back side of the protein so students can identify them. So we have these models of like the potassium channel, sodium potassium pump. We also have the GLUT carrier protein, Aquaporins, and a sodium channel. So the classroom kit actually comes with all these variety of molecules, the potassium and sodium ions, little water molecules, and glucose molecules. So you can show and demonstrate the movement of all of these different molecules across the cell membrane. Yeah, thank you, Ryan. Yeah, it is really cool to be able to show this. Okay, so these transport proteins are going to be helping substances move across, particularly things that are polar because they're repelled by those phospholipid tails. And so there are some proteins that form kind of a tube-like channel and allows this passageway for different ions to cross and for water as well. So you can see a variety of channel proteins here. The carrier proteins are going to actually bind to and interact with these molecules to help transport them across the cell. Some of them will transport without energy required, so with passive transport. Others require some energy in the form of ATP or GTP. And so we have these pumps, active transport pumps, so sodium and potassium, and then the proton pump. So these are the two different types of transport proteins. So you can see here in this diagram that channel proteins provide this channel or passageway, which are specific to the types of molecules they let across. Some of them are not as specific and can let, anything as long as it's a certain size or certain charge to cross the cell membrane. And then carrier proteins actually bind to the molecules they transport, and the protein itself changes shape and then releases those molecules to the other side. So we'll actually be showing you how you can model both of these types of proteins using these kits. Okay, so passive transport. This is a graphic showing different examples. You have simple diffusion, non-specific transporters where you have facilitated diffusion. So that's using a protein to help certain molecules get across. You have carrier proteins, and the third one, and then the fourth one is just an example of a channel protein, which is an aquaporin, and we'll talk about aquaporins in just a little bit. But all of these are moving substances from an area of a high concentration to low concentration down a gradient. Okay, so let's talk about aquaporins. These are one of my favorite proteins to talk about. They're found in every cell type. There's, I think, 13 different aquaporin types. So I'm going to show my model here on camera. Here's a model of an aquaporin protein. And you can see in the graphic as well, a schematic diagram. So this is kind of fun because, and this I purchased from 3D Molecular Designs, and the students love handling this. And one of the fun things about this model is it shows all the different 3D features like the alpha helices. And so these help to anchor this protein across the membrane. And then there's magnetic parts, so you can actually remove a couple of the helices and see on the inside that there's water molecules in a single file line going through it. So it helps give students sort of a more visual picture, tactile picture of what's happening inside this three-dimensional protein. So because the graphic is this cutaway of a 2D image, but students really need to understand that if you're holding this protein, you can see the water molecules going through a center pore right there. So these aquaporins transport water and other small uncharged molecules and helps to maintain that homeostatic balance of water inside and outside a cell. And so it's bidirectional, water can go in or out. And these water molecules go through in a single file line. And so I have couple of other images of ways to show proteins. We have the ribbon model on the left, and then this one is with showing all the different atoms involved right there. And all of these images you can pull from the protein data bank. And so it's really great to show students, besides in a blob sort of picture, that this is scientific representations, several other models of showing proteins. All right, so this is a great animation for students to watch, showing a cutaway section of the aquaporin and how water can move. So you can see that They're going single file through the center channel area. And for the AP level students, you can talk about how as the proteins are in this 3D shape, they're folded in a way because of side chains, these R groups coming from these amino acids, and they participate in chemical interactions with these water molecules to help move water across. So I'll show that again. That's kind of fun to see. Okay, so let's go ahead to our kit now, and you can show water crossing a cell membrane. So this is with the classroom kit with the yellow cell membrane there, and then you can find the foam Aquaporin and put that in there, so it can show that. So this is the classroom kit, but I'm going to overlay here in my camera and show the student modeling pack here. So here is Aquaporin right there. So I think it's taped it. It's got the sticker on the back, so they stick right on there onto this diagram. And then you can plop in some of your water molecules, both inside and outside of the cell, to be able to show movement of water across the aquaporin. So, have students just kind of moving them across. And so that's the setup. And then now we're going to talk about some different scenarios of different concentrations of water on different sides of the cell membrane. Okay, so this is where you bring in the terms that we typically use in biology for describing the amount of solutes on one side of the cell or another. So we have hypertonic solutions, isotonic, and hypotonic. And it's really important when you're teaching these terms to make sure that students understand that we're referring to the solute concentration. So it's whatever's dissolved within water when we're using these terms that have tonic on it. And then in AP, we also discuss water potential, but that's for a different lesson. So this will be helpful for students to be able to see the actual solutes and kind of look at the differences between one side and the other. Okay, so you could have students model hypertonic solutions. And so you could take your sodium ions and throw them into your model too. And you can show that hypertonic solutions have more sodium ions concentrated outside of the cell than inside. So that's why it's hyper, more sodium on the outside. And then isotonic solutions, you could have students start to move those sodium ions as well as the water molecules to show that they have equal concentrations of sodium ions outside and inside for isotonic solutions. And then for hypotonic solutions, hypo means lower or less than. And so on the outside of the cell, you have fewer of those little sodium molecules or ions, and then a lot more on the inside of the cell. So we're describing here the solution outside of the cell. And so now you can have students think about, in terms of the water concentration, where is there a higher concentration or higher water potential, and which direction will water move in each of these cases? So it's a great way for students to see, oh, well, for hypertonic solutions, water will tend to move out of the cell from where there's a high concentration or high water potential to where there's lower water concentration outside or lower water potential. Isotonic solutions, water will flow in and out, and so there will be no net change. And so water pretty much will stay in terms of volume on the same sides that they started off with. And then hypotonic solutions, water is actually going to go into the cell to where there is a higher concentration of solutes in the cell. And so in the first case, you have cells are going to shrink or shrivel. Isotonic solutions, cells maintain their shape. And in hypotonic solutions, cells will fill with water and will swell up, and depending whether they're animal cells or plant cells, they may burst or lyse or just be turgid. That would be plant cells. So I like this model because you can set up an example in the front of the classroom where you could have stations and have students talk about where is the solutes on the outside or inside, and then which direction will water move, and using the terminology. I think it's pretty confusing for students, and it helps them to see it and to reinforce these ideas in multiple ways. Okay, so what I like to do is, after describing these terms and making sure students understand hyper versus iso versus hypotonic, now let's apply it to some scenarios. So case study number 1 is a patient is admitted to the ER with severe vomiting and diarrhea. They get an IV solution on board to restore fluid loss, but shortly after, the patient experiences a dramatic loss in blood pressure. And they send a sample of the patient's blood to the lab, and they take a look at the blood cells, and a lot of the blood cells appear very swollen. And you can see that, instead of the biconcave, kind of the flat little cells, a lot of them are kind of puffy right there. So in the chat, explain, what do you think happened at the hospital? So in the classroom, I would have students talk about this scenario. Did water leave or enter the cells to cause this blood sample to appear this way? And then using their cell model with the foam kit, what happened and which way did water go? Okay, so Ryan says they accidentally used water instead of saline IV fluid, and cells became hypotonic and water rushed into the cells. Yeah, so this hopefully doesn't happen in hospitals today. They do have different IV bags, some with saline and 0.9% saline, and others that are distilled water. And usually they don't have that available for nurses to just grab out of, their medicine cabinet to hang on the IV pole. Usually it's only available in the pharmacy for compounding purposes. But they have to be really careful about the labeling on the bag so that nurses don't accidentally, or folks don't accidentally grab the wrong bag of IV solution and administer it to patients. So using the model, students can show, oh, well, what might have happened, as Ryan suggested, is that maybe they used pure water instead of saline, which has salt in it, or sodium and chloride ions. And so students could show that on the outside of the cell, there's this influx of pure water, very low amounts of ions there. And then inside the cell, which does contain different materials, and it could show, students can show that there's a lot more sodium ions there in the inside. So the inside of the cell is hypertonic compared to the outside, which is hypotonic. And so water will flow into the cell, leading the cells to swell and then burst. So that would be a really bad situation right there. So I did read a case that had happened in, I think, 2003. There's miscommunication between the doctor and the nurse thought that the doctor was ordering distilled water to be administered. And the patient began to lose blood pressure. And by the time they caught that and took out the IV, it was already too late. That patient actually died. So definitely labeling on the bag is important. And then for students to understand why it's a bad idea for students who are at parties, instead of drinking alcohol, that you can actually drink water and drink to death in that case. And I think that's a really good example there. Case study number 2, this is helping students to see there are different scenarios of thinking about concentrations of materials inside and outside the cell. So case study number 2, a couple takes their 7-year-old son to the doctor. The child complains of being thirsty all the time and getting up several times during the night to urinate. And when they do the testing, they find high levels of glucose in the patient's urine and suspect that this patient may have type 1 diabetes. So now students probably can discuss about folks in their life, maybe siblings or maybe themselves or grandparent or friends who have diabetes, and it can help them understand what is going on, why diabetics can experience frequent urination and increased thirst. So in this, you can use glucose molecules in this case to show what's happening outside of the cell, and that with excess glucose being excreted by the kidneys, that water follows and water will leave the cell. And so you have increased urine production there. So really another great application case right there. Okay, so other proteins that you could use to model include sodium channel. So you could talk about the flow of sodium across the cell membrane. Since it's a charged molecule, it will be repelled by those hydrophobic tails. So it requires the help of another channel protein for sodium to cross. There's a couple of images that I grabbed off of the protein data bank here. And then 3D Molecular Designs also has physical models of the sodium channel. And you can, when you tilt it so that you see down the channel, you can see how large this hole is right here. And you can imagine sodium ions going into this channel of this protein. So in the student models, you can see that you have here in the camera, you could have the sodium channels and have students demonstrate sodium ions moving through the sodium channels. You could also have students demonstrate gated potassium channels. This is also in the student modeling pack. And so I've got this example down here, this gated channel right there. I'm going to go ahead and also show this on the classroom kit right here. For this one, the potassium ions can travel through this particular protein, and due to the environmental signals, the cell can be triggered to open up these gates, and so they physically actually change shape. As you can see here in the diagram, there's a closed position and an open position for this channel. So when it's open and triggered by cellular signals, potassium ions can flow through from areas, again, of high to low concentration. So this is still passive transport. You can also show the transport of glucose. So in the student modeling pack, there's also the glucose carrier protein now. So the modeling pack for students has these glucose molecules shown like this, okay? And then in the actual classroom kit, you can see on the screen there, there's the actual foam glucose molecules. So you can show how glucose is transported into a cell and why, if a person is unable to trigger the signal to open up these glucose transport carrier proteins, that it can lead to high levels of glucose accumulating on the outside of the cell, as is the cause of diabetes. And so that's related to insulin receptor issues, but that connects with the glucose carrier proteins, which brings in the glucose. So great examples of a variety of different molecules that you can transport across the cell membrane. Okay, so now let's do active transport. And if you have your modeling kits with you, this is where we can show the movement of sodium and potassium through sodium-potassium pumps. So anything that is called a pump, as in this graphic, we got sodium and potassium pump, calcium pump, or proton pump. They require the use of ATP or GTP as energy sources to be able to change the shape of these proteins to allow for molecules to go across. And so this is an example, these are examples of active transport where you're moving molecules from areas of low concentration to high against their concentration gradient. And so the best example to show is the sodium potassium pump, which are found in all of your cells. And they help to facilitate nerve signal transmission with neurons. And so this kit is great because we have the sodium and potassium pump here. And so I'm going to demo this on the camera. Students get really confused with all the different steps of what happens. And so the first thing you want to do is make sure that you have high sodium concentration on the outside, and then lower potassium concentration there on the outside, and then higher potassium inside and lower sodium concentration inside. So there's an imbalance where one side has more sodiums and one side has more potassiums. And so now this is where students can see if you're trying to move sodiums out, there's already a lot of them out there. So you're going from low to high concentration. And potassiums, if you're moving them from outside to inside, you're also going against the concentration gradient. And so you can walk students through this process that you're seeing this difference in concentration. And within the cells through cellular respiration, producing lots of ATPs. So in my model right here on camera, I have an ATP molecule, and students can see with this student modeling kit version, you've got some paper ADP and a phosphate group, and you can see how they can come apart to be separate and then together to make ATP. So with the 3 phosphates there. And so that's our energy source needed to be able to power this particular pump. All right, so here's the steps. So I've got the graphic on the screen, but in the classroom, what I like to do is to walk through it with students. And so if you have your kit at home, you want to start your protein with these little swinging parts, so they're facing the inside of the cell. And then because of that, we can load up our sodium ions first. So there's 3 little notches, and you can load up the sodiums there. And then there's a portion of this protein you can see where there's kind of a docking site for a molecule of ATP. So you can dock that to that part of the protein right there. And then when there's hydrolysis performed and ATP is broken down into ADP and the phosphate group, this phosphate group is left attached to the protein, which allows for the protein to change shape, and these little parts swing to the outside. And now the sodium ions can come out of the protein and go into this extracellular fluid, which is more protein, more sodium ions, and then load in the potassiums. And then when the phosphate group is removed, these little swinging parts then face to the inside of the cell, and the potassium falls out, comes to the inside of the cell. So we've transported both sodium and potassium against their gradients. Sodiums are being pumped out, potassiums are being pumped in right there. So this is a great kit to use also if you teach anatomy with neuron action there. And I like to use this also as we talk about cell signaling and ATP and nerve transmission. So you can use it for a variety of different types of examples of lessons or units here. Okay, last thing we're going to talk about is a protein that is not in the kit, but you always want to bring real life examples of transport proteins for students. So this is kind of a bonus lesson to be able to include. And because this kit doesn't have chloride ions, it's easy to solve. You can find Play-Doh, or in my case, I just took some paper and cut them into another shape like triangles and put them put chloride, Cl- on there. So these represent chloride ions. You also want to sprinkle some water in there in your cell, water molecules in and some water molecules out. And then set up the stage for talking about the Cftr protein. Right. And so I'm using this, this is a potassium, gated potassium protein, but it resembles how the Cftr protein works. So let's talk about that. The Cftr protein stands for Cystic Fibrosis Transmembrane Conductance Regulator Protein, and it's located on chromosome 7. And the Cftr protein is fairly large, 1480 amino acids, and it's a gated chloride ion channel. So it acts like a gate similar to this model right here, where there are parts that swing open and allow chloride ions to go through. So what can happen is that when ATP binds to this protein, it allows this protein to change shape and then open up and chloride ions to go through. And this particular protein's involved in cells mainly with production of sweat. So on epithelial cells on the surface of the skin and lining pretty much all sorts of organs, digestive fluids and mucus. So lining the epithelium of the lungs as well and air passageways. So this is a graphic showing the placement of the Cftr protein. It's a transmembrane protein, and the parts that kind of swing open are facing the inside of the cell. So this protein is coded by DNA in the nucleus, and then it's produced outside on the ribosomes. And there's a whole pathway where there's the production and then they're trafficked to the cell membrane. And so you can see this graphic here. Chromosome 7 is in the nucleus, and it's transcribed into the mRNA for CFTR. And that mRNA is used in the cytosol or cytoplasm, where they're read by ribosomes. The protein is produced or synthesized in the endoplasmic reticulum. And then there is a folding and attachment of carbohydrate groups. And then they're trafficked to the Golgi and then hopefully to the surface of the cell membrane, where you can see in number 9, step 9 right there. So let's go ahead and model the role of Cftr protein to transport chloride ions and subsequently water across the cell membrane. So my camera view shows something similar to this. So we've got the Cftr protein, can represent it with one foam piece. And then you also want to put in an aquaporin there as well to show that water can move across the cell through these water channels. And I also wanted to show, just as an example, I actually have a 3D printer in my, my classroom, and I found a file for the Cftr protein and downloaded it from the protein data bank and then transferred it to be able to print it. So this model that you're seeing on the camera is actually a 3D printed protein here. And it's great because you can see a little bit through the center of it how there's kind of a open channel for this protein, for these chloride ions to be able to go through. So I can also put it right here in my model to demonstrate that's where that would go. So this is what can happen is as chloride ions are transported out of these cells, notably epithelial cells, once the chloride ions go to the outside of the cell, water will follow. And so when that area outside the cell becomes more hypertonic and the water potential is lower, water leaves the cell and goes into that extracellular fluid area outside of the cell. And so this is the basis of making that fluid-like mucus that coats airways, for example, and the production of sweat. You have water secreted from your cells, and it has to do with these chloride ions. And so when water diffuses out of the cell, if you're talking about cells that line the air passages, it helps to thin the mucus which is useful, as we're going to see in the next video right here.
Cystic fibrosis, or CF, is a genetic disease that affects multiple organs, including your lungs, and limits your ability to breathe over time. Your main airway, called the trachea, branches into each of your lungs. The largest airway within each lung is called a bronchus, and each bronchus branches into smaller airways called bronchioles. Normally, airways are coated inside with a thin layer of fluid and mucus. The mucus catches dust and germs from the air you breathe in. Then, hair-like projections on the airway surface, called cilia, push the mucus, trap dust, and germs out of the lungs. In cystic fibrosis, the mucus is so thick and sticky that the cilia can't push it out of your lungs. This thick, sticky mucus can build up, trap germs like bacteria, and block your airways. Because of this, you may periodically have acute infections or exacerbations of the disease, causing symptoms such as increased coughing, production of mucus, and shortness of breath. As the disease progresses, long-term damage can occur in your lungs. This happens when germs, like bacteria, get into your lungs and cause chronic infections and inflammation. But there's a lot you can do to control the infections and slow the progression of lung damage. Talk to your CF care team about how the different parts of your treatment plan can help keep you healthier. As your partner, they can help you find ways to fit your treatments into your daily care routine.
There we go. Okay, so as you can see, it's really important to be able to produce this mucus on the surface of epithelial cells that line respiratory tract and air passages, so that the cilia can actually sweep away the mucus and reduce, pollutants and infections and microbes and things like that. But the problem is you can have mutations in some of these really important genes that code for proteins. So this protein is the Cftr protein, which is coded for by the Cftr gene. And there so far has been identified over 2,000 different types of mutations that can occur in this gene. The most common one is called the F508del, and it occurs in about 90% of the cases of cystic fibrosis in the US. So this coding, if you're not familiar with this, is F is phenylalanine, which is in position 508. So it's the amino acid that should be number 508. But because of the genetic mutation, the entire 3 nucleotides that code for this amino acid are deleted from the Cftr gene. And so this amino acid is completely missing from the protein that's produced. So what can happen is this protein is not even trafficked to the surface. And so that is a problem. Some of the other mutations, I'm going to show a graphic in just a little bit, have mutations in different places, and which is when doctors can identify what type of specific mutation has occurred in this gene, there is some good news for some of these CF patients. So if you have mutations in both of your copies of your Cftr gene, what results is you're unable to transport the chloride ions. So the secretions, the mucus secretions become really thick. Mostly it's the lungs that are affected, but also other organs which are lined with epithelial cells can be affected as well, like the pancreas, liver, kidneys, and intestine. And one way to be able to diagnose a person who has cystic fibrosis is to do a sweat test and to find that there's high salt concentrations in that sweat. There's also other signs and symptoms, as you can read here. And a lot of newborns are tested at birth with genetic testing. I know here in Colorado, both of my daughters had that little heel prick blood test to test for the most common genetic disorders, and CF or cystic fibrosis is one of them. Cystic fibrosis is most common among people of Northern European ancestry. So it's estimated that 1 in 25 Americans or people of Northern European ancestry are carriers. Incidentally, if you do the 23andMe genetic test, you can choose to do the health test there, and it can reveal whether you are a carrier for cystic fibrosis or not. So there's a lot of individuals who do this 23andMe testing and actually find out they are carriers and didn't know it before. And it has happened with one of my friends. And so this mutation is least common in Africans and Asians, and the average life expectancy is about 42 to 50 years of age in the developed world. So these are some of the possible mutations that can occur in this particular protein. Mutations can lead to proteins that don't even get fully produced. And so they never actually leave the endoplasmic reticulum and ever get trafficked to the surface. Some have issues being folded, and you have mutated versions that get embedded in the cell membrane. Other ones make it there, but they don't function very well. So very little chloride ions are actually transported across. And so there's different degrees of severity of cystic fibrosis, this disease. And so when a patient is identified to have cystic fibrosis, they undergo genetic testing to find out what type of mutation there is. And so class 2 that we're seeing here in this diagram is about 88% of the people who have at least one mutation. And down below mentions some small molecules that can help to correct or augment the function of this protein. And so we're going to talk about this because this is what scientists are studying, the structure of these membrane proteins to be able to help individuals with mutations. So we can model this with students and talk about some of the different reasons for severity or problems with this protein. And so you could have students show that the non-functional Cftr protein shows that there's a lot of chloride ions trapped in the cell. And so because they're not being transported across, water is not going to move across out of the cell into that area where mucus normally would be to thin those mucosal secretions. And then if you have a completely missing Cftr protein, you can see here and show students like that's not even there. It didn't even get made and embedded into the cell membrane. And so again, you have chloride ions being collected inside the cell there. And so if there's less water on the outside, the mucus is going to be thicker and really difficult to sweep out of airway passages. So this is a graphic showing that there are actually other membrane proteins involved, including sodium ion transport. This is more complex and bigger scale, but for students, you can just talk about chloride ion transport and its role in those mucus secretions. So the good news is if you can understand the different mutations and what it leads to, and there's different classes or severities of what can occur, on this graphic shows some potential molecules that could help correct this problem. So there are ones that are called potentiators or stabilizers. These potentiators are molecules that can, in terms of these drugs, they interact with this protein that is something just a little bit is not functioning correctly. And with the addition of that molecule helps this protein to be able to function correctly and move chloride ions across. Stabilizers help with the folding of the protein. So perhaps it doesn't fold correctly and even make it to the surface. And then some of the other, it's really difficult to be able to, for the proteins that don't even get fully made and they get degraded and never make it to the surface, it's harder to find treatment options for those patients. But there's some talk about using CRISPR-Cas9 gene editing technology that still is at work and being developed. Let's take a look at a potential drug solution, drug treatment option.
A patient with cystic fibrosis is susceptible to infections of the lungs. This is due to a mutation that affects a protein on the surface of lung cells. Many different mutations can cause cystic fibrosis, but all of them involve a transmembrane protein called Cftr that functions as a channel for chloride to move in and out of cells. In this case, Cftr is less able to transfer chloride ions out of the cell compared to the functioning protein in people without cystic fibrosis. The channel is, in effect, locked shut. This inability to transfer chloride ions has an effect on the outside of the cell. In people without cystic fibrosis, there is a balance of chloride ions inside and outside lung cells, which maintains the right amount of water and salt ions in the mucus lining the airways. Cilia normally sweep the watery mucus from the lung lining, keeping the surface clean. In patients with cystic fibrosis, chloride ions concentrate inside cells and draw in water by osmosis. With less water, the mucus becomes thicker and cilia can't move. Flow is reduced, providing an environment for bacterial infection. But there are now drugs that can help patients. To find a drug, researchers tested over 200,000 small molecules to see if any would bind to the Cftr protein. Then, for each small molecule that had some binding ability, researchers chemically synthesized different variations of that molecule and tested them for the ability to improve the function of some types of mutant Cftr channels. They identified a drug that, when taken by patients with a certain type of Cftr mutation, binds to the Cftr protein, opening the channel and allowing improved ion transfer. This increases chloride ion flow and maintains a watery, clear mucus, which helps the cilia move more freely and reduces the risk of bacterial infection.
Okay, so one of these small molecules has been found and actually being used by cystic fibrosis patients with a certain type of mutation. So individuals with a G551d mutation, so this is a substitution where there is a different amino acid in that place, means that the folding of this particular protein is just slightly off. And ivacaftor, also called Kalydeco, has been useful to help restore some functionality of this particular protein. And even it hasn't restored 100% function, but it is enough to be able to have patients report that it is making a significant difference in their ability to have a normal life. And so this drug is on the market. It's being used by, I think, about 1,000 people. I was trying to do the math here across the United States. Unfortunately, this mutation, the G551d, is not the most common one. And so there's only a certain limited number of patients who are able to be eligible for this particular treatment. But the exciting thing is, as scientists study the various mutations and be able to find the drugs that can help to restore function or the production or folding of these particular proteins. So that's a little bit more than what a freshman biology class probably would want to know, but definitely at the AP level, which is where I teach, understanding protein structure and function, especially with transporting materials across the cell membrane, is really important. And my students really get hooked into understanding this and why it's important for scientists to study these proteins and be able to find solutions or drugs or treatments for patients who suffer from these diseases. All right, so this brings us to the end of my presentation. And I just wanted to highlight again that the student modeling packs that you received or are going to sign up for and receive in the mail soon are the packs on the right. And then on the left is the classroom kits. And that what you're seeing there is for a one group set. So in my classroom, I actually have 6 sets of these. So I have one for each of my tables, and each large table of students of 4 to 6 students can utilize that and model that together. So I would welcome any questions that you have. Feel free to ask in the chat, and it looks like we have about 10 minutes. And Heather's got some links to these different materials as well.
Well, while we're waiting for any questions in the chat, I just want to thank you, Chris, for a great session. Good question. How do you clean the models between student use? I know Heather has a resource of what we've used. Just because the foam is a closed cell foam, you can spray it with just any disinfectant spray. Chris, what do you use in your classroom?
Well, at this time, since COVID transmission doesn't seem to happen with touch surfaces, I just have my students make sure they wash their hands before and after using it, and it's been fine.
Great. So just a couple of reminders. Please join us tomorrow when Chris presents using models to make connections between DNA and chromosomes. That'll be tomorrow at 12:45. Also, she used the phospholipid membrane transport kit and the membrane student modeling pack today, as well as she showed the Aquaporin channel mini model, and I didn't put it on here, but the sodium channel also is available. And make sure that you use the discount code. It's CASE21 for 20% off most of our products. You will receive a session evaluation after this session, so please go ahead and complete that. That helps us to determine what sessions we present in the future. And there will also be some information for the CBM and their summer courses. I believe that's how Chris came to Milwaukee and did a summer course. And so that is a very valuable use of your time if you ever want to come check out Milwaukee, Wisconsin. We have some great things. Hopefully we'll be in person again. And we're happy to answer any other questions you might have. Got about 6-7 more minutes.
I just want to say one thing, if you're still hanging on here, is I actually just used this kit 2 weeks ago to teach about cells and cell membrane and transport. And my students actually told me, hey, we love these kits. It's hands-on. We actually like them better than doing labs in your class. And so if that's anything, that's an endorsement for the, that students really enjoy being able to model with these kits, and it helps them to understand better. They get, I guess, too stressed out doing the labs in my class.
Thanks, Chris. That was a great session, and that is what we hear from a lot of teachers, is that their students are often happy when they're in the hands-on materials, and particularly the foam-based kits. The foam materials tend to make everybody happy.
I also want to, I don't know that I said it out loud, but you can also borrow kits from the Msoe lending library. And so there's a link at the bottom here. And I think maybe Heather put it in the chat, or if she hasn't, if she will, there it goes.
Right now.
Yeah, perfect. So you can also borrow kits from Msoe's lending library. You would go online and request a kit. They'll ship it to you FedEx, Heather, or UPS. I'm not sure.
I think they may ship out FedEx, and you can return it however you want.
Yep, and all you would pay for would be return shipping. So it's a good opportunity. You can rent it for 2 weeks, use it in your classroom, and see how your students respond to utilizing those. There's also some, I think we have some tips of ways to get funded for additional kits. If you borrow them and then think, oh, I would really like to have a set for my classroom, there's some other ideas there. And if you have any questions or anything else, you will stay on as long as you'd like. You can also feel free to unmute yourself and turn on your camera if you'd like. Feel free to leave whenever you're ready as well, but we'll stick around and answer any questions anyone might have.
And we can probably go ahead and stop the recording now, Kim.